Thank
Great.
you so much, John, for organizing this workshop. As someone who studies E. coli, I thought this has been extremely educational for me to not learn not just learn the background for SYN development, but also for mycoplasma biology in general. So I'm going to be like Ji-Wong, where I'm going to tell you about a method I'm trying to develop, and it's still sort of raw at the moment. And I would appreciate any sort of technical feedback that you all could give. My name is Josh. I'm a grad student in Jay Shao's lab, and I'm from the Johns Hopkins School of Medicine. So, as we all have seen several times by now, from developing Syn 1.0 to Syn 3.0, you can see that it went from having a very small and homogeneous phenotype to one where you had cells that are both small and large, and they took longer to divide on average, from 63 minutes to 178 minutes in this original paper. And just earlier this year, James Pelletier and Li Ji Sun from Elizabeth Strakowski's lab were able to identify, if you were to look at gene clusters in the Syn 1.0 genome, one particular cluster that when you knocked it out produced cells that looked very similar to Syn 3.0. Here's a pointer there. And if you compare that to adding that back into the genome to produce Syn3A, the cells were a lot more homogeneous again, similar to 1.0. And this is just quantifying the area of cellular form, showing that shift in cell size. So what's very interesting among the 19 genes that they added back in were that there were some very essential genes that were necessary for producing that shape. Included among those are those responsible or implicated in cell division like FTSZ. You've just heard a talk from Ji-Wong on doing some super resolution work and investigating more about what the cell division specific genes are doing and how they might be organized. On my end, I'm very interested in the growth dynamics of these cells. In particular, I want to see if I can establish some sort of method that can quantify the growth of Syn3A in real time. And realistically, this would mean that I have to track the cell size and shape of individual cells. And this would be helpful because then we can compare growth states of various Syn varieties as genes are added back in or subtracted, or whatever minimal components you might mix around in your media. You can see how that affects individual cells. And ultimately, we're doing this in collaboration with the Luthi Schulten group, in particular with Maddie Stover's talk, who talked last week, for them to develop a kinetic model for cell growth. Now, I'm approaching this similar to how my lab has done it when we've imaged E. coli. That is, if you can look at a phase contrast image of a single cell and then track that cell's growth, you can see how one cell has to elongate through a certain period of time, then divide into 2 cells, then 4, then 8, etc. And through phase contrast microscopy, you can automatically segment those images and track that change in volume through time. And if you were to perturb that in any particular way, like say adding antibiotics to those cells, you can see how they filament and die and have a really sick looking phenotype compared to the wild type. So wanting to take this agarose pad based approach where we prep our cells, I figured this would be a nice way to begin, simply because an agarose pad can help immobilize your cells and have it provide reliable places that you can return to for time-lapse imaging for several hours. And so what this would involve would essentially just applying some molten agarose with your media of choice, letting that solidify, and then mixing your cell sample with that agarose pad and covering with the cover slip. In our lab, we use something called an FCS2 chamber, which is, instead of using slides to produce your agarose pad, we've got a gasket in there that seals your agarose pad and your bacteria, and so that you can exchange media or buffers that you would like. In our case, we're using this because you can also interface it with an environmental controller to heat your stage up to 37°C so that you can try to provide an optimal environment for these cells' growth. So when I first started trying to make this method, I thought, okay, I'm going to make it similar to E. coli. Make a dense agarose pad that's 3%, centrifuge my cells with a hard spin and throw them on the scope at room temperature. What happens? They don't grow. So this was when I first realized that, bacteria across multiple groups, of course, have different biology. So in this initial image, you can see that there's absolutely no growth. And in some cases, you can see cells actively lysing through this 13-hour total movie. So I had to do a little bit of optimization, identify one variable at a time. These are the top 3 that I affected. And by the summer, I was able to identify some conditions, such as if you bring down the agarose pad percentage and reduce the pressure that's applying to the cells, you bring the stage up to 37C, and you don't centrifuge the cells and you treat them carefully, you do start seeing clusters of cells grow. In this case, you can see individual cells scattered throughout the fields of view, but you don't largely see those dividing for whatever reason. But you do get reliable reproduction of clump growth, and you can zoom in on them a little bit further with these 5-micron scale bars and see how the area of the clump form can grow. Through some more optimization, because 0.25% agarose pad is nice and it helped, but it was too liquid. And this condition, while I could find growing cells, I also would very frequently find what I would think be a good cluster of cells, and within 2 frames, they would fall out of focus and diffuse away, just because it was so liquid that they weren't staying in one place. So after a little bit more optimization, I have landed on a condition where I can at least reproducibly find some clusters now, where I brought my agarose pad percentage up to just a little bit, 0.5%, and I can gently centrifuge my cells to concentrate them, but still treat them somewhat gently. And now I can at least see clusters of cells grow. And you can see some select time points in this image where once every 30 minutes imaging, you can see that the cluster is definitely growing. The only issue at this point is that I cannot identify and isolate individual cells and track them. I'm limited to these clusters at every single point of time. So we turn to imaging them expressing mCherry as well, in the hope that maybe we can use this to help isolate where the single cells are and use that to track them through time. But right now, this is just where the method has gotten. So from this, I have identified some key next steps that I plan to do. So first of all, now that I've finally gotten, I think, a condition to reproducibly make them grow, I'm going to be working on the automated software that's tracking the cells. So making the autofocus more reliable, and the tracking and the mCherry localizations between each frame more reliable. Then I need to work on how I can individually look at these individual cells through time. All of the kind of quantification that I have done so far is just more or less following the growth of the area of these cells, which you can see that does correlate, and you can see some sort of growth, but the problem is that it's not tied to individual cells. This doubling time that I'm getting is far too long compared to what would be optimal for Syn3A. And the hope is that if I can, be more consistent in, for example, the autofocus, then perhaps maybe the integrated intensity of these images might be a more trustworthy measurement at some point. But because the focal plane kind of varies between frames, it doesn't ultimately help tell me any information at this time. So anyway, this is where the method is at the moment. I want to thank my lab, in particular Jay, for being a great mentor, as well as our lab, or our collaborators like the Luthi Schulten group that is helping produce this kinetic model. JCVI, in our collaborating meetings, they've been very helpful in helping me learn and understand how these cells work, as well as how to properly grow and work with them. And then, of course, the Hall lab as well. And in particular, Ji Wong, he and I are constantly messaging each other about how to grow and work with our cells. So anyway, I'll take any questions if you have them.
Thanks, Joshua. I was wondering, what do you centrifuge your cells in?
SP4 medium. Well, like centrifuge them in, as in the medium they use?
Yeah, in the medium, are they suspended in a buffer?
No. So in order to watch these cells in real time, I'm trying to treat them as gently as I can. I'm using SP4 medium that's warmed up to 37C to resuspend them. I'm taking the media that's culturing with them, and I centrifuge that down, resuspend in fresh SP4, put them on an SP4KO agarose pad. They're constantly with their medium the whole time.
This is SP4 with knockout as opposed to SP4 with FBS.
Yes. SP4 knockout.
A quick question. I saw that on the slides that you had before, you were showing your mCherry technique. I saw like these little and I know that this is probably not what it is. I was just really interested how you see like on like the side, for example, like below like that, like timestamp on your right slide, it looks like
Mhm.
they look really similar to mycoplasma colonies when you look at them under like an optical microscope. Like for example, that one that's like directly to the
Mhm.
left of that zoom in right there. Do you know what those objects are? No, the one above that. Sorry, like they look like a
Oh,
fried.
this one?
Yeah,
So that is an out of focus cell. And this is what I was saying. My problem with these
yeah.
0.25% agarose pads is that my cells would be very diffusive and at very wildly different focal planes. And if you were to change that, that would be more or less a normal cell. It's just way out of focus.
It's a very
It's a
interesting
good
technique,
question.
though.
Yeah.
Josh, if we could make your cells to put a protein on the surface that would let them attach to glass, could you just grow them in liquid media, do you think?
I've thought about that. The only concern I have is that sometimes, like for example, like looking at E. coli and adhering them to a cover slip, it does affect their physiology to some extent. I don't know how it would affect Syn3A, but it definitely would make things more well behaved. It's worth a shot.
Does anyone have any thoughts about how we might make that work? Well, not today. Other questions for Josh? I know we're running a little behind. Did anyone have any thought? I asked a group question about making the cells not clump. Does anyone have any experience with that? How that might be a better way to have him enable Josh to get single cells?
Actually, yeah, I'd be curious to see that. I'll say in one of my optimization days I've done before, I took my cells and I put them in like a sonication bath to see if I could like break up clumps. I ended up still had some clumps after I didn't want to over sonicate them, and none of them grew because I think I must have hurt them.
We had done stuff with Mycoplasma genitalium and pneumoniae where we treated the cells with 5% glycerol and passed them through a 0.1 micron filter, but that seems not to work as well for Mycoplasma capricolum and mycoides. And 10% glycerol killed the cells in the case of genitalium and pneumoniae.
Interesting.
And that might work on Syn3B, John. This is James Dobbins.
Yes.
That might work on Syn3B because it doesn't have the capsule. That may be what's interfering with the mycoides performance of Syn1. It makes, when I say a plethora, that's an understatement by far. 90% of the biomass from an mycoides culture is that galactone capsule. And you can get rid of that with glycosidase, and it would probably pass for a 0.2. I think Syn3 would probably pass for a 0.2 micron filter. We also did some work with sonication and trying to get these organisms to exist as a single cell. And it's going to be difficult, honestly. One thing, I don't really think that M. genitalium, Mycoplasma genitalium, which another species, but I don't think it exists as a single cell at all in its life cycle.
Well, you see them, James, you see them gliding on surfaces, single cells.
Well, that's an artificial inducement. I don't think in a natural environment or in vitro growth, they ever exist as a single cell.
I guess my question is,
we have several papers that look at sonication in pneumonia. You can find them. I think the primary author is Art Cotton.
Could you post those in the chat, please, James?
Yeah, I can find them and put them up. John, would you do the honors?
Want me to send Art's paper on that?
Yeah, that one. You know which one.
Yeah. All right, I'll go pull it up. Give me a minute.
You might try a little of really low concentrations of non-ionic detergents. Clean 20 comes to mind. I mean, really low level concentrations, like 0.01% or something. After sonication, to get them to exist as a single cell for a little while and function.
It's really helpful, at least to consider and look into. Thanks for the paper in the chat.
Hopefully that'll be of some help. I'm sure Art would love to hear that.
The summary is basically that it's dependent on time and level of sonication.
Mm-hmm.
And you do walk a fine line between killing them and separating them.